Hi everyone. So I hope you have a good coffee break. Um, Hello, my name is Jonathan Clavijo. I'm a member of the Plant Science Council and also a PhD candidate at the Horticultural Science Department at the University of Florida. Thank you for being with us today and welcome back to the sixth annual Plant Science Symposium. It's my pleasure today to introduce to you to our student speakers. First, we will have Edgar Sierra. Edgar was born and raised in Mexico. He obtained his bachelor's degree in plant science from the Universidad Autónoma of Chapingo in Mexico. Upon graduation in 2009, he was hired as a pest and disease manager for a 36-acre farm producing bell peppers, blueberries, and blackberries. After working there for 7 years, in 2016, he obtained the Fulbright Award and moved to Florida to pursue his master's degree at the University of Florida. He graduated with a Master in Horticultural Sciences in December of 2018. Since January 2019, he has been working towards his PhD, also at the University of Florida. He is a student in the tomato breeding program at the Gulf Coast Research and Education Center of the University of Florida. The focus of his research is the identification of resistance to targets of tomato caused by the Corinospora cassicola in tomato wild relatives, also the characterization and mapping of major resistant genes, and advancement of resistance into cultivated tomato. Just wanted to remind you that this is part of the section of the presentations of student speakers. We will have 4 in total. And today, Edgar is going to present us his presentation entitled "Breeding for Resistance of Target Spotted Tomato: A New Threat to the Tomato Industry." Welcome, Edgar.
Thank you for the great introduction. I'm very happy to be here today. Thank you for inviting me. As Jonathan said, my name is Edgar. I am a PhD student at the Gulf Coast Research and Education Center. I am starting my 4th year and I work with target spot of tomato. So today I'm going to be presenting you a little bit of my research with the theme breeding for resistance to target spot of tomato, which is considered a new threat for the tomato industry. Okay, let's move on. I'm going to start by saying a little bit about the tomato industry. So it's a big industry in the United States. Florida is, from that, is the 2nd producer of fresh market tomato, which accounts with a little bit over 1/3 of the production. And together with California, they have about 2/3 of the national production. So the threat is called target spot of tomato, which is caused by a fungus Corinospora cassicola. This one is a necrotrophic ascomycete with no known sexual state. It is widely distributed in the tropics, requires high humidity and warm temperatures, and also free water for infection. And it infects lots of plants, over 500 species. So this fungus was first reported in Florida in 1967. So it's been around for a while, but it's been it's been a disease since then. It's not that it hasn't happened before, but just recently has become very problematic to this southeastern area. So why is it a threat? Well, if no control is implemented, it can be very destructive. Actually, it can be one of the most destructive foliar diseases in southern Florida. The second point is that it will infect foliage and fruit, so it can have infections in both. And it can also be confused with other diseases, so its effect might be downgraded because of that. For instance, you have target spot here in one picture, then early blight, and then bacterial spot. All of them are necrotrophic spots in the leaves, so a grower can easily say, oh, that's bacterial spot, or that's early blight, while in reality it might be target spot as well. So in addition to that, given the climate patterns right now that have been changing and have been becoming warmer, and then shifts in rain patterns and stuff, we can have also some outbreaks in other areas, or the problem can become even worse. Some of the symptoms, all of them are necrotrophic spots, lesions, or plaques. So in the foliar area, we have lesions that can develop very big and have a target-like appearance because of the fungal growth. And in the fruit, those will be flakes that will also get wider and also have necrotrophic, fungal growth in there as a target-like appearance. For the foliar, it can result in defoliation, and for the fruit, it's very problematic because even the smallest flakes will cause your fruit to be not marketable, so you cannot sell it anymore. So that's the disease. So how do growers control it right now? So they only use fungicides to control target spot. But recently, there has been some papers documenting, and also the growers have been aware that the fungicides don't work as well anymore. And the FRAC has listed this pathogen as having a high risk to develop resistance to fungicides. There is no known resistant tomato cultivar, so we cannot rely in resistance right now for that. And it seems like previously wood breeding was not of interest because, as I said, it's been just recently that this has become a big problem for the industry. This is the only paper that I found documenting some resistance, screening for resistance in tomato accessions or variety, and it dates back to 1973. That's everything we have for resistance. So given all of these, the goals of these projects, or the project that I'm working on, is first to identify resistance to target spot in wild tomato relatives, characterize and map major resistance genes in QTLs, and advance resistance into cultivated tomato cultivars. So before I present the results, I'm going to be talking about seedling inoculation assays for a few slides. So this is what I mean by seedling inoculation assays. So we have about 6-week-old plants in pots. When they are at this age, we spray inoculated with a cocktail of Corynespora cassicula isolates. We put them in a growth room for 4 days with high humidity, controlled temperature, and controlled light. And after the 4 days, we take them out and rate disease in the foliage. So this is the seedling inoculation assays. So the first step, as I said, was to try to identify resistance to target spot in wild tomato accessions. So for this, we had a set of 24 Solanum accessions, and we included 3 susceptible controls. We did the seedling inoculation assay that I told you before, and we did 6 different experiments. So we tested 1 isolate of Corynespora cassicula per experiment. So we had a total of 6 isolates. The design was an RCBD with 16 blocks, and the data was analyzed with a linear mixed model. So I combined all the data of the 6 experiments into a single analysis. Very quickly, this is the average rating for each of the 6 isolates. So in the x-axis, you have the different isolates. Each data point is the average within each experiment for each of the accessions that we tested. And the scale, the Y scale, is the average rating, and it's inverted. So you will have the most resistant accessions at the top. And of course, the susceptible controls at the bottom that were expected to be infected, to be very, to be full of disease. So they are. So this is basically the main take-home message of this. So as I said, we combine this data of the 6 experiment into a single mixed model analysis. And I calculated the LS means, and this is what we obtained. So all of the accessions had some level of resistance compared to the susceptible controls. So we could find some level of resistance, but what we wanted was the topmost. Even though they were not significantly different from each other, we wanted to select a set of representative accessions from the 3 different species tested to follow resistance. So we selected 2 Galapagensis, 1 Chismanii, and 1 Pimpinellifolium accession. So I'm going to start by showing you what we did for the 2093 accession, which is a Pimpinellifolium. So for this one, we actually had access to a recombinant inbred line population that also included markers, so that was very nice. So it was a total of 125 reels coming from the 2093 by NC EBR1 cross. So these reels, we did a seedling assay in those ones. We put them in a 13 replicate incomplete block designs because we had so many reels we couldn't replicate each one of them because there were too many. It was not feasible. So we did at least 6 drugs per reel. So we did the experiment in the seedling assay. We calculated the LS mean from the model, and together with the market data, we used it to do inclusive composite interval mapping analysis. The top picture shows you 3 resistant reels together with the resistant parent. The bottom one is 2 susceptible reels together with a susceptible control. So we did this experiment twice, and this is what we obtained. So a single QTL in chromosome 12 in both experiments was associated with resistance to target spot. And in both cases, they explained at least 30% of the variation. So that was very nice. So we kind of were done with the QTL mapping with this accession. So now we moved on to the two Galapagenses, which were the 483 and 532. For these two, we used a different approach. So we had F2 populations coming from the cross from each of those to 8059, so 312 plants for the 483 derived population and over 500 for the 532 population. Seedling inoculation assays again, and we extracted DNA, and from there we followed the QTL-seq approach suggested by Takagi et al. And I don't have much time to explain you how it works, but if you're interested, you can email me and I'll show you how this works. But basically, this is what we obtained. So the top panel is the 483 population, the lower panel is the 532, 12 different chromosomes, and the black line will show you the associated regions of the genome with the phenotype that I was giving. So this is really not as clean as we expected it to be for a number of reasons that, again, I don't really have a lot of time to discuss, but basically you are having a wild accession cross to a cultivated one, so there is some differences in there, and then you are rating foliar disease, which can come also from other contamination with bacteria or other things. So we use this panel just to select those pigs that were suspicious to have a QTL. So we designed markers for a few regions in the genome for each of the population, and we did QTL mapping with those. And this is what we obtained. So we were able to map QTLs in chromosome 4, 7, and 12 for the 483 derived population, and 3, 8, 9, and 12 for the 532 derived population. So that was really good. We were able to get some QTLs out of here in an F2 population. So moving to the next generations of backcrosses. So we had backcross 1 F2s and backcross 2 F2s for the two different accessions, so 483 and 532. And we did the seedling assays, run markers and everything. And now we were able to obtain QTLs now in chromosome 9 and chromosome 12. Chromosome 9 for the 532 population, and in chromosome 12 in all of the populations that we were testing. So we were down to 2 QTLs now. That was for the seedling assays. Now we wanted to try the resistance in the field. For this, we tried combinations of genotypes at two different QTLs. So for 483 accession, we tested combinations of chromosome 4 and 12. 4 was significant in an analysis of variants that we did, but not in the QTL mapping. So we wanted to see if there was some effect in the field. 532, we had combinations of 9 and 12. And 2093, we had just the QTL in chromosome 12 that we mapped previously. So in total, this gave us 21 different genotypes that we tested, and we added a few controls. So all of these, we have them in a randomized complete block design with 6 reps in 6 plant plots. Excuse me. And after planting, we did 2 foliar inoculations 6 weeks after planting with a Corneaspora coccicola cocktail with at least a log 4 concentration of spores per milliliter. And we did 7 foliar ratings from which we calculated the AUDPC. So we were really curious to see how the resistance held up in the field. We were not sure if the seedling resistance would translate into the field, but we were so excited and happy to see that actually it translated to the field. So here in the pictures, you have susceptible on the left and resistant on the right genotypes for the chromosome 12 allele or chromosome 12 QTL coming from the 2093 accession. So you can see here that clearly there is something going on, or something that is having some good resistance in the plants. And basically this is the AUDPC calculated after the 7 ratings that I did in the field. So we have in the x-axis 27 different genotypes. The 20 to 21 are the genotypes we tested. The rest are just controls. For now, we're just interested in the target spot susceptible control, which is the one here I list. And for example, here, the 1 to 9 are combinations from within the 483 background, chromosome 4 and 12. So the brackets show the groupings of the susceptible, heterozygous, and resistant 412. And within each one, we have susceptible, heterozygous, resistant for 4, and same here, same here. So these first 9 are for 483, coming from 483. The middle ones are coming from 532, and these are combinations, again, susceptible, heterozygous, resistance coming from chromosome 12, and then chromosome 9 in each of them. So susceptible, heterozygous, resistant for chromosome 9. Finally, the 2093, you can see the effects even better. You can see it more clear. And what we can basically see from here is that it seems that chromosome 12 seems to be the strongest or the most clear QTL that will provide resistance to target spot of tomato. So that was very good. That was very exciting. So the next thing I did, I didn't want to do all the pairwise comparisons because there were too many. So I wanted to test compare within each background the resistant genotype against the susceptible here, and same here against susceptible against resistant, and same thing, and then resistant against the target spot susceptible control, and finally all the resistant genotypes between them, but just for chromosome 12. This is what we obtained from those contrasts. So the chromosome 12 resistant genotype is significantly different from the susceptible control. So all of these genotypes are susceptible, are significantly different from this control. And also within each background, they are significantly different to the susceptible genotype as well. And finally, the 483 genotype, resistant genotype, seems to be significantly better than the other two. So this genotype in chromosome 12 coming from 483 seems to be significantly different from this and from this. So we are breeding, right? So now we are also interested in, okay, how is this going to affect my fruit size? So for now, we really don't want it to go into depth in that because we just have backcross 2 and backcross 3. And you see the fruit is still very big for the domesticated tomatoes. So we don't really know yet, but we can tell that, for example, if you just compare chromosome 12 here, these 3 here, you don't see a big difference. And also here, you don't see a big difference in fruit size. So we are confident that when we get closer to this level of fruit size, we won't have a big difference in fruit size, which is good for growers as well on the breeding purpose. Some future work that we have going on is we have a field experiment again this season, and then we're going to do a third field experiment where we are going to measure also horticultural traits. We plan to begin fine mapping of these QTLs, and also, if you didn't notice, we didn't do anything about the 524 accession, which is a Chismeni eye. So we are planning to begin fine mapping on those as well. So thank you to all these amazing people from the tomato breeding lab and also the plant pathology lab at GCREC, and thanks to my committee members for all the support. And thanks to you all for your attention. And here's my email in case you have any other questions or anything to follow. Thank you.
Thank you so much, Edgar. A really nice presentation. I just want to remind to our audience that if you have any questions, please use the Q&A section so you can formulate your questions for our speakers. So we have some time. Edgar, could you please explain us a little bit about the QTL-SEG approach? What is that?
Yes, so in this approach, basically, you will have your population segregating for a given trait, and then you will have the lowest, in this case, we are talking about disease, the smallest ratings and the highest ratings in the plants. You will have the DNA from those plants, you will bulk it together, purify, and send it to sequencing. So you will have readings sequencing for 2 bulks, we can call it the resistant bulk and the susceptible bulk. So that will be DNA from a bunch of plants that are resistant and a bunch of plants that are susceptible. You send it for sequencing, and then you do all the alignment work, and you will calculate the SNP index that you will be kind of subtracting between them, so you can have a curve. So if you don't have any difference in genotype or in allele frequency, you will have basically a delta index of 0 because there is no differences. But whenever you are selecting for a given region for each of the bulks, then you will have peaks. That's where you expect your QTL to be. That's in like a broad definition, but if anybody has any more questions about that, I can just send you the paper where they explain in detail how this works.
Thank you, Edgar. So also you mentioned something about, I think it was in slide 8, not sure. It was one slide where you have plenty of graphs. you mentioned that your results were not as clear as you expected.
Right.
Why you think is that?
Yeah, so this one. So this is the QTL-seq. So ideally, you will have all of your chromosomes to be like this, and then just a single peak like this one here, where you will see, oh, this is a QTL. But you can see there is like huge regions of the genome that appear to be significant. That is for a number of reasons, could be for a number of reasons. First of all, we are taking just samples, like in this case, from 558, I took 20 resistant plants and 20 susceptible DNA. So that will give you some bias. And then you have also a cross between a wild tomato and a domesticated one. And then you are expecting to have some differences in the genome, small difference that could be affecting your alignment and your SNP calling, which will also affect this. The third reason is that you are dealing with a foliar necrotrophic disease. So if you have some Xanthomonas perforans around, and then you put the plants into high humidity, high temperature, you will not only have a target spot, you will have other diseases as well. So that will create some variation. And then if that variation translates into the plants that you are selecting to do the QTL-seq, then you will actually have some QTLs that are false positive in this one. So basically here, as I said, what we used this for was just to do to select the regions to target for QTL mapping, basically.
Perfect, Edgar. Thank you so much for your presentation.